Finally, after what, 5 chapters and what seems like 6 months, we get to the point of the semester where we actually get to talk about something alive, don't we? Right? We haven't been talking about anything alive yet. We've been talking about molecules. Are molecules alive? Are atoms alive? Yeah, they are. Water's alive, are? Yeah, no. Atoms aren't alive. Molecules are not alive. Now, when do these molecules get together in such a way to be alive? Because I've told you before, the living things are nothing but a chemistry, right? We're chemical reactions. That's why we spent the last 5 chapters doing chemical reactions. That's all right, no big deal. The idea is we had to lay the establishment for the life is tied to chemistry. It's tied to physics. And so if we understand chemical reactions and we understand physical reactions, physics and such, then we understand life, because life is nothing more than a personification. That's not a bad word. A personification of these very specialized chemical reactions. Metabolism, which we'll move into in the next few chapters, respiration and photosynthesis, are some very specialized, some very complicated reactions. So it's not just simple, sodium and chlorine sticking together doing electromagnetic attraction. So I'm excited now because we get to move out of the realm of the foundation of the things that you have to know. You have to know the chemistry. You have to know some of the basics before we can actually talk about something alive. And right here in this chapter 6, we get to that thing that's alive, because we're going to talk about something in the next slide known as the cell doctrine or the cell theory. Now, you guys know what a theory is. We've talked about a theory. A theory is not something that's just easily given out or easily defined, right? We don't say, well, that's a theory. Theories have to be things that have been tested through several hypotheses, right? Having gone through the scientific method a bunch of times, and before they become a theory. So think about a theory as a set of hypotheses or a promoted idea. So you come up with an idea and you test it and so on and so forth. And then you eventually say, I've seen this enough times that I can make a theory, or I can state a theory that the Earth revolves on its axis once every, it's not exactly, but pretty close to about every 24 hours. So once you start being able to time hours, you can substantiate that theory over and over and over again, right? So a theory, when folks say it's just a theory, you understand now that that's not quite understanding what a theory is. So let's look at the cell theory or the cell doctrine. And the first one talks to exactly what I was just getting to, right? All living things are made up of cells. Oh, not the first one, I'm sorry, the second one. And cells are the smallest thing we get to that are alive. All right, so all living things, oh wait, go back to the first one for a second. All living things are made up of cells. You guys did relatively well on this in the exam, so I was very impressed. Deductive or inductive? All living things are made up of cells. All living things. Think about, Which way is the logic flowing? Which way is the reason flowing? Are we going from big to little or little to big? We say something like all living things are made up of cells, we're making a very wide statement. We're not saying I've looked at cell 1 and cell 2 and cell 3 and all these things are living, so therefore if you're living you must have cells. That would be looking at a bunch of little things and building it up. So Colin, you got it. Yeah, it's inductive reasoning, right? All living things are made up of cells, therefore if we want to, if something has cells, it must be a living thing. The logic is flowing down. When you make broad proclamations, therefore all living things are made up of cells. Okay? Any questions? You did better on the exam than that. Any confusions as to why the logic's flowing from big to little? All planets revolve around their star, their sun, in the same way the Earth does. And we know that because we've looked at the Earth and Venus and Mars, and now we've looked at other planets out there, and they're all doing the same thing. So that's a very inductive, a very wide statement. Okay? Makes sense? Problems, concerns, confusions? Okay. All right. All right, so we say that cells are the smallest thing that have all the characteristics of life. So that means they're organized, they metabolize, they evolve, they reproduce, which is what the next one says, and they respond, right? All those characteristics of life that we talked about don't come together yet until we get to the cell. They don't occur in proteins, they don't occur in carbs, they don't occur in all that stuff we did in the last chapter. It's not till we get in Bio 2, I talk about one of the big jumps going from non-living to living was when we began to package very specialized chemical reactions like metabolism, like respiration. And the cell is that package, okay? Now, the last one in this cell theory or cell doctrine is actually kind of an interesting one because it poses an awful lot more questions than it answers. For example, it says that all cells, well, not for example, it says that all cells on Earth, there it is again, there's that inductive part, all cells on Earth come from pre-existing cells through something called cell division, right? We'll talk about cell division, I don't remember what, mitosis, okay? And meiosis, actually. You can see it here. Some cells, for example, some organisms are nothing but a cell. They're what, unicellular? So, for example, this critter that you see in here, look at the scientific name, Saccharomyces cerevisiae. What's the common name of this critter? You might be thinking, how in the heck would I know? Look at the species name, the second name. Or what do you think? Well, no, if you knew what we use it for, then you probably know what it is. If you speak Spanish, perhaps you might know what this is talking about. What's a cerveza? Important word to know if you ever go to, spring break to Mexico, I guess. Isn't a cerveza a beer? Yeah, it's beer. There you go. So this word right here, Saccharomyces cerevisiae, remember when you name something, that second word, you can kind of make it whatever you want. Sometimes you name it after somebody. Sometimes you have a little fun with it. And you say, oh, you know what? We make beer with this thing. I wonder if it was a Spanish fella, Spanish-speaking person who discovered Saccharomyces cerevisiae. I mean, when you discover a new species, you get to name it. So that's interesting. Okay. But yes, this is yeast. That's what you're looking at. And yeast reproduce through something known as asexual reproduction. It's not very exciting. It's simply just splitting, or sometimes it's called budding. If you look on this one here, there's a lot of budding going on. Obviously, there's one bud about ready to separate there. There's one on eye. There's one there, and then there's a bigger one down there. That's what yeast do, right? They reproduce asexually. Okay, so that's cell division for them, but it's also reproduction. We replace our cells. Our cells divide, which we'll talk about, but we don't reproduce when our cells divide. We just get bigger or replace a scar, right? Things like that. Okay, so y'all happy with that cell theory or cell doctrine? All living things are made up of cells. They have all the characteristics of life, and they come from other cells. Does not particularly answer the question fully about what the first cell came from. Okay, and again, that's something outside the scope of this class, but it is something I do discuss in Bio 2. And if you're interested in that, what you're talking about is something called biogenesis. You can take a look at it online. And there's some very interesting and compelling arguments to suggest that we could move from the chemical to, or simply the non-living chemical to the living chemical, if you think about it. So interesting stuff to think about. All right, now cells. Let's talk one thing that we all are familiar with about cells. We looked at a paramecium under the microscope. How big were those paramecium again? Do y'all remember? How long were they? You guys are pretty good at calculating sizes. Nobody has their nobody has their lab book near them. Were they 66? Yeah, I think they were. Is it 66 or is that the length? Yeah, that's the length of them. That's right. 66, I think. Why am I drawing a blank? Anyway, that's small, right? That's small. I mean, whatever it was. Oh, by the way, it's not millimeters. It was microns. Yeah, I think that was the width. Yeah, it was around 40 in width. I can't remember. I want to say about 100 or so, or 200. Yeah, it was about 200, wasn't it? About 200 microns in length. Okay. Regardless, and we don't need to waste any more time about this. You can look in your field or you look in your lab books, but that's small, right? That's not big. I mean, you needed a microscope to see it. And when you looked at the slide of the paramecium, you might have seen little red dots, maybe, if they were gathered together. But holy cow, I mean, that's not very big. So there's a question to be asked there, right? If all cells come from pre-existing cells, that means early life, the yeast are alive, okay, there's the fungus. This early life was small. It was a cell. And when it made other living things, it reproduced asexually, and then things evolved from there. But where I'm getting with this is when the decision to go from microscopic life you need a microscope to see, to macroscopic life, big life, us, right? There was a physics constrained, mathematically constrained fork in the road. If you think, what the heck is he talking about? All right, here's the choice. Once we go from little cell or a little organism to big organism, there were two ways it could have been done. One, little cells could have gotten bigger. Okay, so maybe like you might see a paramecium hanging out at the mall, or macroscopic organisms became multicellular. Now you know the answer to the question, right? But do you ever think about why? Why did it happen that way? That's the beauty of science to me. Beauty of science is we look at that and we go, wow, there was two ways, but we don't see the other way. In no instances, I mean, I think the largest single cell is 750 microns. It's a bacterial cell. So what, maybe 3 times, almost 4 times. You can see it with the naked eye. It ain't very big, but you can see it. It's a sulfur bacteria. Okay, but cells are generally small, so there must be a reason for that, right? We can take a look at this scale right here, and we see that if we were talking about, what do we say, 200 microns? So we're talking about paramecium, and this is in a logarithmic scale, so it doesn't, the scale may be a little unfamiliar, but 200 microns is probably about right here where the human egg size is. Right, and they're not very big, and even if we go 750 microns, we're still 1 millimeter would be 1,000 microns. We're still, kind of over in here, smaller than a frog egg if you've ever seen one of those, right? Chicken egg is obviously too big. So anyway, and again, unaided eye, so it's macroscopic. So back to the original question. When organisms decided to become macroscopic, they obviously went multicellular, right? We have trillion cells in our body. 10 times as many prokaryotes, by the way, live in and on our body, just so you know. But there's about 1 trillion cells in our body. So obviously multicellularity, and that's a very interesting evolutionary trait, by the way. But anyway, multicellularity was the choice. Not a great word. But anyway, multicellularity was the result. There must be a chemical and or physical reason why that happened. So let's take a look at it. Let's do a little bit of math. And I'm going to remind you back to lab number 1 when we measured the blocks. And I know we got a little confused because I had you switch units and I shouldn't have had you do that. That was my fault. But if you remember, the concept of the blocks was as we measured the blocks, as the blocks got smaller, from the big one to the little one, what increased? Do you remember? Do you remember what increased as the blocks got smaller? It was a value we calculated. It was actually a ratio we calculated. Do you all remember what that was? It was related to the volume R. It was related to the volume, and it was related to the surface area. Remember, you measured the outside of the block all around 6 times. Well, you have to measure it 6, and then you took the calculations. You did the volume as well, and you put those two together in a ratio. Now, do you remember as the blocks got smaller, what happened to that? We abbreviated it in class, or I did it up on the board like this. What happened to that surface area to volume ratio as the blocks got smaller? So the blocks are getting smaller. Does the surface area to volume ratio stay the same, go up, or does it go down? as the blocks got smaller. Yep. So it's an example not of a direct relationship, not the surface area, and it's a surface area to volume ratio. Okay, you got to make sure it's that quantity that you're talking about. Yeah. So with the surface area to volume ratio, it goes up as the blocks get smaller. That's a physics, those are wooden blocks that we measured. Those aren't cells. But let's think about that for a second. That means a small block has more surface area per unit of volume. I don't remember exactly the numbers, but if I recall, I think the small block had something like 3.2 square millimeters of surface area per unit of volume, per 1 cubic millimeter of volume. Okay, the math aside, as the blocks got smaller, there was more surface area around them per unit. Now, cells have to communicate with their environment, don't they? Right? They've got to be able to get rid of waste products, and they've got to bring in food. And regardless of how you look at that, whenever a message, be it a chemical message, an auditory or oral message or visual message is being relayed, it has to be moved from point A to point B. Now, in the case of a cell, it just happens to go across the membrane, thank goodness, right? For reasons that we'll talk about in subsequent chapters. All right, so if you have a lot of membrane, if you have more membrane per unit of volume, then you don't have to be as concerned with jamming up things moving across the membrane. It's like a traffic jam, all right? The smaller an item gets, the smaller a cell gets, the more surface area it has, the more space it has to move things across its membrane as dictated by its volume. The bigger the cell is going to need more things, the smaller the cell needs fewer things. And since it has that higher amount of surface area, that membrane to it, it actually is more efficient. So it is just physics, folks. If I took those blocks, those wooden blocks, and I turned them into metal blocks of the same size, iron or something, and I heated them up all to the same temperature, hot, like, I don't know what, I think iron melts at 2,000 degrees Fahrenheit, whatever, really hot. And I set them aside, I heat them up to the same temperature, boom, and then turn them off all at the same time. Which one would cool off faster? Now, maybe you did this in physics, or maybe you're thinking about what I just said about surface area to volume ratio and what it does. Which of those blocks would cool off faster? Which one could you grab first, the small or the large or the medium? If you had physics, I know you talked about this. You had to have, because it's a basic principle. Hold on, folks, I got my contact. If you type something, I can't read it. Okay. All right, which one cools off? Well, at least make a guess. Well, that's a guess, right? Yeah. Sorry, Sadie, not the large one. Yeah, it's the small one. It's the small one for the same reason. It's for the same reason that a small cell has more surface area to move things across, waste going out and nutrients and oxygen coming in and carbon dioxide going out. And later on, we're going to talk about synthesized proteins that go out, whatever we're talking about that has to be communicated between the cell and the environment. That's what it's doing when you bring up oxygen. You're communicating something, a message is being transmitted. It just happens to be chemical. When you're small, you have more surface area. Now, back to the blocks, the smaller one's going to cool off faster because of the fact that it has more outside surface area per unit of volume. So there's more space per unit of volume for stuff to for the cooling off to occur. Okay? You got to remember that, yes, the bigger block, the bigger cell has more membrane, but it also, the volume increases a lot faster. Take a look here in this example. Now, up in the upper left is what I'm trying to get at. This is a good thing to put in your notes. Cells remain microscopic because they try to make that surface area to volume ratio as big as they possibly can. For the reasons I just explained, right? They're more efficient. So, don't worry about the formulas for surface area of a sphere and volume and all that. But if we took a small cell and a big cell and differed them by 10 units, whatever that is, skip all the stuff in the middle, but look at the surface area to volume ratio. Look what it does. just increasing it by 10 units, 10 millimeters, 10 microns, we decrease its surface area to volume ratio by what? A tenth? Right? 0.3 is a tenth of 3. Isn't that something? I love that, right? Isn't that cool? So we take physics, we take an iron block cooling off, and that's how scientists do things like that. They go, okay, well, maybe the physicists understood about the iron block and understood why it cooled off faster. So a biologist was like, cells are small and they have to move with the environment and communicate with it. Oh, maybe it's the same reason. It's all the interdisciplinary stuff. That's why, why do I have to take all these? That's why you have to take all these other classes, even if you're not a science major. That's why you have to go take humanities and English, so you learn something about the world, and for goodness sakes, you learn how to communicate, okay? All right, so cells are small because it maximizes their efficiency. It makes them more efficient. They're better at interacting with the environment. This is a small cell. This is Bacillus coagulans, which is a prokaryotic cell. It's a bacteria. TEM means transmission electron microscope. That's how they got this shot. SEM is scanning electron microscope. I couldn't honestly tell you the difference in terms of why you would use one or the other, but I do know there are differences between them. All right, this is a typical bacterial cell, a typical rod shape. Bacteria come in rods, they come in round shape, and they also come in spiral shape. So this is your typical rod-shaped bacteria, but there's not much to a prokaryotic cell. So let's talk about the parts of a prokaryotic cell first, and then we'll actually turn our attention after that and talk about a eukaryotic cell. Okay, and you'll see in just a minute what I mean about a prokaryotic and a eukaryotic. And you're thinking, gosh, what are those words he's saying? Here we go. Prokaryotic. Prokaryotic means literally something. Pro is a root that means, let's say, initial. Okay, as in first. And Kary equals nucleus. And you might see that root a couple more times this semester, so that might be a good one to remember. So prokaryotic, or a prokaryotic cell, this is a prokaryotic cell, is an example of a cell with an initial nucleus. Well, it's not exactly the definition, just kind of hints to the definition. If you look on here, look on this chart, you'll not see something labeled a nucleus, do you? Do you see a nucleus on here? I see something that almost says that, but I don't see a nucleus, right? Yeah, there's no nucleus. So initial nucleus, yeah, I see a nucleoid. A nucleoid, you see what it's pointing to? It's actually got a big bracket around it. It's pointing to the bacterial chromosome. Bacteria have one chromosome. Okay, they just have one chromosome. So that one chromosome, like chromosomes are in a nucleus, right? Bacterial chromosomes, that one they have, the reason why it looks like that is it's just a big loop. You could unwind it. It would just be like a hula hoop of DNA. There is no nucleus. So the nucleoid is not a thing. It's an area. Oh, it's like the shells. There you go. It's like shells again. Remember we talked about shells and we said that shells don't really exist, right? Shells are collections of orbitals. All right, so a nucleoid doesn't exist. It's just wherever the one chromosome that the bacteria have exists. There's no nucleus in a prokaryotic cell. The other word I said to you, eukaryotic. EU, when you see it in science, means true, as in it exists. In other words, eukaryotic cells do have a nucleus, but I'm getting ahead of myself there. Let's not skip over the prokaryotic cell. They look relatively simple, right? We like to say ancestral. Better word. That was kind of subjective. But they do have some parts. They have ribosomes, for example, and the ribosomes are involved in protein synthesis. We'll talk more about that later. We'll talk more about that in this chapter, and we'll talk more about that in the later on the semester. So ribosomes are a part, and they construct proteins. This is where the amino acids are linked together through dehydration synthesis, right? This is where you start linking those together to form those polypeptides that we've been talking about. They have a cell wall. It's not, we talked about that. It's not, I think I mentioned a cell wall in plants because we were discussing cellulose. This is a cell wall, but it's not made of cellulose. It performs many of the same functions that the cell wall on a plant does, but it's not made of the same material, okay? Some prokaryotic cells have an additional part on the outside of the cell wall called a capsule, and that's where they have some of this nefarious activity that goes on. What they can do with a capsule, it can be like a slime layer, they basically can hide. The way your immune system knows that a cell is not supposed to be there is there are receptors on these foreign cells, like this bacterial cell, that your immune system recognizes being pathogenic, potentially disease-causing, pathogenic. It says, oh, these are bad cells, so it then alerts the immune system and it attacks. So what bacterial cells do to hide is they actually will cover those receptors in slime. So it's kind of like trying to be invisible, right? It's not really invisible, but it's kind of like the kid who puts the sheet over his head, says, and hide and seek, can't see me. It works among the immune system many times, okay? The flagella, which we'll talk more about, but flagella are used for movement. Many of these things have the characteristic of motility, the ability to move around. Plasma membrane, we'll talk a lot more in the next chapter. We have a plasma membrane as eukaryotes. Plasma membrane is also the cell membrane. Prokaryotes have it too. Fimbriae, these are cool. Fimbriae are the little projections, not the same as I was talking about a second ago with the immune system, but the fimbriae are these little projections that come out by which they can grab onto things. They can stick to things. Like, for example, the ones that might attack your tonsils, they can use these fimbriae to actually attach to your tonsil. And then actually, I just talked about this in my Bio 2 class yesterday, two of them can get together. They can grow their fimbriae and form a tube and pass genetic information back and forth. Very weird. Very weird. Okay? So that's a prokaryotic cell. As I said, they're not simple. I always remember my zoo professor in college. You know, they do so well with so little. They do a doggone good job of being alive. They're around long before us and probably long after us, and live in much more harsh conditions than we do. This is an interesting bacterial cell. It's got a nucleoid, or prokaryotic cell. The cytoplasm is the liquid filling of a cell. It's kind of where all the parts are kind of floating in. There's the cell wall, the plasma membrane. What's interesting about this picture, though, are these photosynthetic membranes down here. We normally just think about plants as photosynthesizing, but man, lots of prokaryotes, lots of these ancestral cells can photosynthesize. We'll talk about photosynthesis in more detail later. It's one of those really specialized chemical reactions, but photosynthesis and respiration, when we talk about it, all depend on a membrane. What happens in both of those is you're going to build up a concentration of something on one side of a membrane. Membranes in biology become very important. We just talked about them in the surface area to volume ratio. So it's the same kind of idea. Membranes are very important for moving things across the cell, but also for causing reactions to occur. Okay, we'll talk a lot more about that later on, right? So I just wanted to show you that even in these very simple cells, we have those that can do these relatively complex reactions like photosynthesis. And when we talk about that in the chapter, yeah, you'll see what I mean by complex. Okay, any questions about prokaryotic cells? Oh, good. Not much to them. They're good at what they do. Some of them could do some really relatively advanced things, and there's lots of them. Let's turn our attention now to a typical eukaryotic cell. So now right away you see the big purple thing in the middle. It's a nucleus. Now we're going to talk about each of these parts, but what's being labeled here, we have a word for them. We don't just simply call them parts now. It's called organelles. These are all called organelles. It's derived or a variant of organ, which are the things that make up you. So the nucleus, the ribosomes in this case, the Golgi apparatus, the lysosomes. This one up here, that's cool, right? Endoplasmic reticulum. All these things that are labeled here, all components of the cytoskeleton, we'll talk about each of those, are all things called organelles. And we're going to go through and talk about each one of these. But again, you notice that this has got a nucleus. And notice, too, inside the nucleus is the chromatin. The chromatin is your chromosomes plus a protein. Your chromosomes are actually wrapped around a protein. Think of it as kind of like packing material, if you will. keep those chromosomes safe. But when they're together, it's called chromatin. Okay. So inside of the nucleus is the chromatin. There's a nucleolus, which we'll talk about. But notice there's an envelope around it, a membrane. That's what it is. There's a membrane around the nucleus. There's a membrane around the cell itself, the plasma membrane. There's a membrane around all these organelles. This one down here, the mitochondria, there's a membrane around it. Membrane, membrane, membrane. All this stuff is membranes. And they're all made of the same kind of membrane. And if you wonder why I keep saying membranes, it's because here, later on as we move through this chapter, we'll learn that because everything's got a membrane around it, and they're all the same kind of membranes, it gives this cell really unique properties. It gives cells really unique properties, not just this cell. Okay? So more about membranes here in just a second. But first, before we actually start talking about the organelles, let me show. This is a typical animal cell. Okay? And this is a typical plant cell. Animal cell, plant cell. I see two big differences here. Plant cell, animal cell. Do you spot them? Actually, I see three big differences. Make that three. Shape. Ah, there you go. What would you call the shape of the plant cell, this one here, more or less regular than maybe this one, the animal cell? More or less regular. Is the plant cell more or less regular? The plant cell? Yeah, I think so, right? Yeah. Yeah, because I mean, this kind of has a squarish type arrangement to it. It's not really a square. It's 1, 2, 3, 4, 5, 6 sides, and they're not. You remember when you looked at the onion cells underneath the microscope, right? They were rectangular in shape, okay, more or less. They weren't all exactly. This one is more blobular in shape. We talked about blobs. So the plant cell has a more definite shape, and that's because the plant cell has something the animal cell does not. It's got a cell wall made of cellulose, those rigid fibers, which the plant needs to stand up, against gravity. There's that physics again. You got to stand up against gravity, so you need a cell wall where these cellulose fibers, these firm, rigid fibers hold it together. Or if you're an animal, you got to have some kind of a skeleton, inside or outside, or some other type. Worms have a different type, but all right. So that's a big difference. Now look at the organelles. Here's the animal cell, okay? I want you to focus your attention on green organelles. And this one, the Golgi apparatus, is probably the closest to green. So now look over here and look at the green organelles. What happens in those green organelles in the plant cell? Any guesses? If you think about it, it's what plants do that we don't. Okay, no, but as far as the green parts, the green organelles that are in this plant cell, these right here, what do they do? So, yeah, they do photosynthesis. That's right, Marie. They actually make food. Now, they do that. They have those chloroplasts. We do not, do we? Animal cells don't. We don't photosynthesize. So that's difference, big difference number two. Cell wall in the plant cell, chloroplast, because they photosynthesize in the plant cell. Animal cell, no cell wall, and we don't photosynthesize, so we don't have any chloroplasts. Okay. Now, well, there's one more minor difference. Let me point it out to you because it does come back later on in our discussions. Animal cells have a centrosome. Okay, we'll talk about what a centrosome is. I was kind of hesitant to introduce it right now, but animal cells have centrosomes. Plant cells do not have centrosomes. All right, so centrosomes. And again, that'll be a little better in context later on in our discussion here. If I learn how to spell it. Centrosome. And then finally, the last big difference, so we got cell wall, we got chloroplast. The last big difference is this thing in the middle that looks like it's transparent. This is a central vacuole. Plant cells have a very prominent central vacuole. Animal cells do not. Now, the big thing that central vacuoles do for plant cells is this is where they store water. All right, so that fill that central vacuole up with water. Now, again, if you're like me, when we were talking about microscopic to macroscopic, immediately you go, well, centrosome's spelled wrong. There's not two O's in it. But anyway, immediately go, why do plant cells have this big storage sac of water and animal cells do not? We both need water. So, you know, why do they have it? Why do the plant cells have that big central vacuole where they can store all that water, whereas we don't? We animal cells do not. Does anybody have a good idea why that is? Think about lifestyle, right? Think about, you know, how plants get water. Think about how animals get water. If an oak tree is too far away from a there you go. Animals can go to the water. Thank you, Sadie. Animals can go to the water to drink, right? Most animals are mobile. They can get up and move to a watering hole, but a plant cannot. Plants stuck in the spot. There's no plants walking around ranging the plains of Africa looking for a watering hole. You know, I was going to say, if an oak tree is too far away from a creek where there's water, but locally around the oak tree there's a drought, unless it's got big roots that have extended all the way out there to that waterway, it's going to drown, even in sight, if the trees have eyes, even in sight of the water, okay? So storing water in plant cells is very important. And you see how the connection's made when these first folks were looking at cells, they probably noticed this big thing in the middle. And you're going to notice it when we look at it under the microscope. Later on, we're going to start looking at Elodia cells. They're up in the upper right. You should recognize the ones in the lower left. There's your cheek cells. Look at these Elodia cells in the upper right. In the Elodia cells, you'll see it too in class. You see these big open spaces. Now, what those big open spaces tend to be is these massively filled water vacuoles. Many times when we're looking at a plant cell, most of the volume inside is that big central vacuole full of water. Most of the other organelles, the nucleus and the endoplasmic reticulum and all that other stuff, get shoved to the corner. Yeah, I'll show you this in lab. You'll see this in lab. We'll do an experiment with this. This is kind of cool. I like this picture. Stem cells being generated by brain cells. So they fluoresce them. They put radioactive fluorescence into the dye, and they use a fluoroscope, I guess, to read the fluorescence in different colors. This is the world's biggest cell. I told you it was a prokaryote, but I forgot about this one, Belonia ventricosa, only because it's actually fairly common. My buddy had it really bad in his aquarium. We oftentimes get it in aquariums. It's a single cell, 5 centimeters in diameter. So it goes really against that whole idea of, but there aren't many of those, right? So there's probably some very special adaptations that Belonia has to deal with surface area to volume ratio. One of them living in the water probably helps. Okay, everybody ready to move on to the actual organelles now? Talk about the different parts. We have any questions? Cool. All right. All right, let's start here with the brains. This is the nucleus. I say the brains because inside the nucleus is the chromatin. That's where the chromosome is. So that controls everything about the cell. That controls everything about protein synthesis, everything how the cell works, right? When we talk about enzymes later on, enzymes digest carbohydrates for energy, but enzymes are proteins. And the code for all of that, the recipe on how to make a protein, are your chromosomes. Contained inside of that nucleus, surrounded by a membrane, a nuclear envelope. Not only just a membrane, but two membranes, an inner membrane and an outer membrane. So it's doubly wrapped, doubly wrapped. You want to be very careful what you let in and out of the nucleus. All right. Now, what's controlled going in and out of the nucleus is controlled by these things called pore complexes, right down here. See that? If you look at it, you can see them in the electron microscope. It's very neat. They're like little rosettes. What it is a series of molecules that can open and close to allow things to move through. So these little rosettes can pinch shut and then open up to allow materials to move through the nucleus. The stuff to move in, stuff to move out, and that's all very strictly regulated. That's why you got these things that can open and close. The nucleus is actually reinforced with a nuclear lamina, or lamina. I don't know why I said it like that. Lamina. It's lamina. And you can see it looks like, oh gosh, I don't know. Fiberglass again, I guess. It looks like it's got some rigidity to it. That's because the nucleus is obviously, again, very important. It's a neat shot. It's cool. Again, it's with a transmission electron microscope. You can see the little pores on it too. It's very neat. Here's what I mean about chromatin. The blue little line that you see there that's being wrapped around those spiky things, the blue line is the DNA. That's the chromosome. And the spiky things are the proteins. The spiky things are actually called histones. So that's the protein. That's the chromatin. Chromatin is the chromosome wrapped around the histones, and we'll talk more about that later on in the semester, okay? So any questions about the nucleus? Notice too, before we go on, notice that there's a connection between this other organelle, the rough ER. ER is endoplasmic reticulum. We'll go over that in a little bit. Yeah, we'll probably get to it today. 9:30, 10:30, 10:50. Right, okay. Notice that they're connected. Right? Notice that the membranes between the rough ER and the nucleus are connected. That's going to become important. All right, no questions on that? Let's jump back for a second to that composite picture. Composite picture means that not all cells will have all these parts, but this would be your hypothetical cell with all the parts. The little dots. Some of you can see them out in the cytoplasm. There's one right there. Some of them you can see connected to that blue thing, the rough ER. Rough ER. That's how you spell. The endoplasmic reticulum is named that because it reticulates, right? Moves back and forth through the cytoplasm. See how it's winding back and forth? The way the artist drew it, looks like it's been cut open. It's actually like a series of these pipes or channels, right? Okay, so those little bumps that I was circling earlier, there are two types of those, and that's why I circled the two types. There are those that are out in the cytoplasm, and those are called unbound or free. And the ones that are attached to the blue part, okay, the ones that are being pointed to on the right-hand side are known as bound. So we got unbound and bound ribosomes, right? The one on the left isn't labeled, but it's an unbound ribosome. Now, regardless of what kind of a ribosome you are, an unbound or a bound one, all ribosomes, and you have them in plant cells too, all ribosomes do the same thing. Ribosomes are where proteins are synthesized, folks. That's where the amino acids are put together. That's where you take amino acid number 1, amino acid number 2, blah, 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 based on the recipe, okay? Now, the ribosomes that are bound to the ER, if you think about what I just showed you in the picture where the ER and the nuclear envelope are connected, the genetic information, which is the code on how to make the proteins, that little connection right there makes it very convenient for the chromosome to send its message out here into the ER to find a ribosome that's attached to it to give it the recipe to make the protein. God, ain't Mother Nature wonderful, right? She puts all this stuff together in the most magnificent ways, in the most convenient way. So here's what a ribosome is. Notice here's a picture of ribosomes that are free and ones that are actually bound, stuck to the ER. I'm looking here. The lines across are the endoplasmic reticulum. We call endoplasmic reticulum either rough or smooth, and the rough is because ribosomes are attached to it, so it looks rough under a microscope. But you can see there are free ribosomes there too. Ribosomes have two parts. They have a large subunit and a small subunit. That'll become important later on in the semester, okay? And there you can see a really cool computer model. You can blow that up a little bit. If you guys can recognize the. Can you see the alpha helices? See that? And it's not quite as apparent, but the beta pleated sheets, right? See how that stuff comes forward? Right? That large subunit and that small subunit had to fit together. So putting those alpha helices, those beta pleated sheets, you know, all that stuff that we were talking about in the shape of a protein and the levels of a protein, you see it evident right there to make the shape of a ribosome. All right. Any questions on ribosomes before we move on to the next part? The next part involves sort of a conglomerate of organelles. Are we good? All right. That's what we're going to talk about next then. Endomembrane system. The endomembrane system means a system of membranes that are linked together. Okay, that's why it's a system that moves out through the inside of the cell, through the endo, which is the inside of the cell. The components of the endomembrane system include the envelope around the nucleus, the endoplasmic reticulum, which I just showed you where they're connected, these other organelles that we're going to get into, including the ER, the Golgi apparatus, the lysosomes, the vacuoles, and then finally the membrane on the outside, the cellular membrane, that membrane that surrounds all of it. Remember I said everything inside of a cell, it's all part of this endomembrane system. All these things have membranes around them. Well, not every part of the cell is part of the endomembrane system, but everything has a membrane around it. And all of these parts are connected together. Now, you're going to see why that's important as we move through this stuff, because it looks like in some of the processes I'm going to explain to you, it looks like the membrane would disappear. And it's because of the endomembrane system that it does not. And we'll talk in just a moment about what a vesicle is. But all these are connected by vesicles, all these parts of the endomembrane system. Now, I've already showed you the nuclear envelope. That's that inner and outer. There's two parts to it. I showed you how it's connected to the endoplasmic reticulum. So, you know, let's go ahead and start from that point. Let's start from the ER. Let's take a look at that organelle. Okay, so we've got, as I said, two types of ER, rough and smooth, and it's all about appearance in this particular case. Rough ER has ribosomes bound to it, and smooth ER does not have ribosomes bound to it. So you can look at that picture on the right. It's actually easier, I think, to see the one down below, electron microscope picture. There's the rough ER. You can see it's got the ribosomes, and the smooth ER does not. Notice that they come together, though. They're connected in some cases. In some cases, it's the vesicles that's going to connect them. All right. Now, up above, I've got some synopsis here of what each part does. The rough ER has the bound ribosomes. They secrete things like glycoproteins. They secrete other types of proteins as well. They distribute these transport vesicles we're going to talk about. And they provide membrane for the cell, for the outside part of the cell. And we'll see how that all works together in just a minute. But notice how it's all connected. Again, the artist has drawn it as if it's been cut open. It's actually a series of these pipes. It's a conduit, things to move across. Lumen, by the way, is the opening in a pipe. So if that's the pipe, then that's the lumen on the inside. In the smooth ERs, where lipids oftentimes get synthesized, where you break down carbs, or at least part of that happens, the smooth ER, especially if we look at liver cells have a lot of smooth ER, and that's because that's where detoxification occurs. Calcium ions, which you need for effective metabolic activities, are stored in the smooth ER. So you know what they do, you know, aside, it's still all part of this endomembrane system. The rough ER is rough ER again because of those ribosomes that are attached are creating proteins that are being dumped inside the lumen, inside those tubes. Okay, so and that those proteins are going to be transported. So let's kind of figure out, as that protein makes its way back and forth across these tubes, it's doing things. Okay, it's already been assembled. So it's making alpha helices. Those hydrogen bonds are coming into effect. It's making beta pleated sheets. It's folding back on itself in the tertiary level. If it's more than one, then more than one are getting together and combining and making a quaternary protein. But all that's happening is that protein is winding its way. That's why that ER is winding back and forth. Finally, when it gets to the end of the ER, that protein gets pinched off. You can see in that red arrow what's going on. You make a little bud, kind of like when we're looking at the yeast, right? And then the transport vesicle gets pinched off. But the transport vesicle is part of the ER. It's just the chunk of it that pinched off. So it's made of the same material that the ER, the same membrane the ER is made up of. and the ER is made up of the same material the nuclear envelope is made up of. Remember, it's all part of that endomembrane system. Now, where's this transport vesicle going to go? Okay, well, this transport vesicle is going to make its way across the cytoplasm of the cell, and here it comes, until it runs into something called the Golgi apparatus. And the Golgi apparatus looks like the ER, although it has similar functions, but is very similar appearance. There are some differences in the function, but notice once again what's happening is that transport vesicle makes its way and touches up against the Golgi apparatus. Right? This is done by chance. They randomly bump into one another, but if you put a lot of them together, then you increase the chance of that happening. Now, the Golgi apparatus under an electron microscope to me always looks like globs of wax from a candle, like when it dries, bloop, bloop, you get the like layering effect. That's kind of what it looks like in the picture. And it does kind of work like that too. But the transport vesicle that's coming from the rough ER attaches itself to the cis face. There's those words cis and trans. A little different context though. We're not talking about we're not talking about geometric isomers here. The cis face is the receiving and the other side is the shipping. But on the cis face, that vesicle from the rough ER, since it's made of the same material as the Golgi apparatus, because the Golgi apparatus is made of membranes, and all membranes are made of the same thing, it melds or blends into it. And the stuff inside of it, those little purple proteins basically, are now inside the Golgi apparatus, where they're further modified, right? Proteins are very important. A lot about shape, so you got to make sure you have the right shaped protein. So you're going to take a lot of time for that to happen. Once that protein is modified, once it's all ready to go, then it's ready to be shipped. So it's made its way through the Golgi down to the transface, and it's pinched off again into another vesicle. So it sorts out these materials, where it's going to go. Some of these vesicles are going to actually be transported or eventually going to transport material out of the cell. Some of these vesicles are going to move off and transport materials around the cell. Right, so that's what the Golgi apparatus is. Sorts that out. It's like the shipping loading dock in a company, right? This package goes here to that department. This package, or the mailroom of a department. Okay, again, all made of the same material. That's the beauty of all made of the same material. That stuff can blend in. It's light dissolves light. Polar dissolves polar. Non-polar dissolves non-polar. It's the same thing here. Since they're made of the same material, they blend in a very liquid form into each other. Okay, now we're going to get to the point where the transport vesicle delivers the material out of the cell. But before I get to that, I want to talk for just a second about those vesicles that are made and stay inside the cell, because a couple of them have a very special function. Now, a vesicle is just a sac, folks. It's just a membranous sac. It's made of membrane material. What goes on in the vesicle, or what the vesicle can do, that's probably a better way of saying it, what the vesicle can do depends on what's inside of it, okay? So the specificity of what the vessel's function is determined by what's inside of it. So let's look at one of these specialized vesicles. This is called a lysosome up there at the top. Y'all remember what lysis means? Yeah, to split. And a some, when you see the word some, like chromosome, lysosome, peroxisome later on, it means something to do with the cell. That's not a very good definition, but a lysosome is something to do with the cell that splits things, okay? Now, first, let me remind you, it's a transport vesicle. That's all it is, okay? So let's take a look down here on the lower left where we're witnessing something called phagocytosis. Now, we'll talk more about that later on, but that literally means cell eating. PHAG is to eat. So what the cell is doing is eating. So what it does is it basically engulfs food. That's what the little brown particles there, into a food vacuole. Now, a food vacuole is a pinch off of the cellular membrane. And since it's a pinch off of the cellular membrane, it's made of the same materials of all membranes, which are all made of the same material. So along comes a lysosome. Along comes a lysosome. And inside that lysosome are things that'll lyse. not the things in your hair, folks. They have digestive enzymes inside. Enzymes that can make big molecules and tear them down into small molecules. And since the lysosome's membrane is the same as the food vacuole's membrane, they merge. The digestive enzymes are then dumped into the food vacuole and the food's digested. Beautiful. Thank goodness those membranes are made of the same thing, right? Now, on the other side, we have an example of something called autophagy. Yourself, to eat yourself, basically. What happens in autophagy is the lysosome that merges, it doesn't merge with a food vacuole in this case, it merges with a vesicle that includes an old worn out organelle. In this case, it's a mitochondria. Right? Think about the cell for a second with all its organelles. Just because an organelle goes bad doesn't mean the cell has to die. You can replace the organelle. It's kind of like in your car. You know, if the tires go flat, you don't need to buy a new car. You can buy new tires. It's much more efficient that way. Organelles and cells, after a while, they're all systems. They fall apart. But, I mean, you can get a number of years of life out of a cell, is what I'm trying to say. But organelles can go bad. And what you do is you recycle them. You digest them back up into principal parts and then reassemble them, okay? So this is what's happening with this mitochondria. And the peroxisome we'll talk about in just a second, if we get to that today. What time is it? We might get, yeah, we'll get to peroxisomes. Peroxisomes contain hydrogen peroxide. Kind of nasty stuff, right? You have a wound, you put hydrogen peroxide on it because it kills many organisms. Well, anyway, the lysosome merges with that mitochondria that's inside that vesicle, all made of the same membranes again, and the hydrogen peroxide and what's inside the lysosomes, which we can just kind of collectively call hydrolytic enzymes, breaks down the mitochondria. Hydrolytic enzymes would be enzymes that engage in hydrolysis, right? The splitting by water. Okay. And sometimes entire cells, or actually, sometimes what happens is lysosomes inside of a cell will all spill out their contents at the same time and actually destroy the cell, kill the cell off. That's one of the things that helps separate here. I'm going to do this on the, when we're first developing, there's tissue between our fingers. Closer, there we go. And later on during development, the tissue is gone. And one of the things that destroys the tissue between the fingers is autophagy of the cells that make up that tissue. So all those cells in there die, and then your fingers come apart. All right, any questions on that? Lysosomes, phagocytosis, or autophagy, two big things we talk about with those. All because of membranes that fit together. Now, let's go with the other type of I'm sorry, I'm jumped ahead of myself. Let's go back to that central vacuole a second. The central vacuole is basically membranes off of the ER and the Golgi apparatus. It's a big, giant membrane. And this central vacuole has lots of functions. It can hold water. If it's storing water, it's called a water vacuole. If it's storing pigment, it's called a pigment vacuole. Poison ivy, or those plants that give off a toxin that irritate you, oftentimes that toxin is stored in these vacuoles and released when you brush up against it. In the paramecium, you see in the upper right, look at these cool things. We'll look at these in lab. These are called contractile vacuoles. Isn't that neat? The one in the middle is filling. The one on the left has squeezed out, and the one on the right is squeezed out further. What are they squeezing out? They're squeezing out water. Paramecium, because of the way they're made, water's always dumping into a paramecium. It's always flowing into a paramecium. So if they don't have some way to get rid of the water, they'll explode. So they use these contractile vacuoles to squeeze. It's really cool to watch too. There's a video on Blackboard you can see. Okay, so these central vacuoles have a variety of functions. The big one that we normally concentrate on, again, is the central vacuole, or the water vacuole in plant cells because of water. Look what I mean about the central vacuoles getting so big. Look at that picture in the lower right. There's the central vacuole. Look, the rest of the cell is crammed up against the edges of the cell. Most of the cell's volume is that vacuole. Isn't that amazing? Okay, back to what I was going to say about transport vesicles moving outside or moving materials outside the cell. Well, you should be surprised if you take a look at this. Transport vesicles come off of the trans side of the Golgi apparatus, and they then fuse with the plasma membrane, like you see that one on the right that's happening. And since all those membranes are made of the same thing, regardless of different colors that the artist is using, they all blend into one another. And if this was, like, say, a secretory vesicle, which just be a vesicle that secretes something, this might be the secretory vesicle that secretes digestive enzymes in your saliva. So the cells of your salivary glands would be producing this purple little dots that you see in the picture there, and secreting those into salivary ducts, which eventually would dump in your mouth and start the digestion process. You actually start digesting starches right inside your mouth before they get to the stomach. Notice in the picture too, everything's all connected, right? It's either connected, directly connected, or connected through transport vesicles. Now, how this answers the question of why doesn't the membrane eventually disappear is because other processes like phagocytosis are actually recycling membrane the other way, bringing it back towards the nucleus. So there are other components of the membrane system that are moving in the opposite. So this is really nothing more than just a cycle of membranous material, all with functions: transport vesicles, phagocytosis. Where are we? Oh, good. All right. The next two slides I'm just going to briefly go over because we're going to talk a lot more about these organelles and respiration and photosynthesis. I'm just going to point out a couple of things about these cells first. This is the organelles. This is the mitochondria, and the mitochondria is the powerhouse of the cell. Okay, it's where energy is converted. It's where food energy is used to make, well, you've heard about it before. It's used to make ATP. ATP is energy currency. This happens in the mitochondria. It doesn't exclusively happen in the mitochondria, but most of it does. Okay, and the details of that I'll discuss later on, but let's go back to the actual anatomy of this thing. It's got an inner membrane space. It's got an outer membrane. It's got an inner membrane. They're all made of the same materials, all membranes. It's actually got its own ribosomes, its own DNA. That's rather curious. Excuse me. Other organelles don't have their own DNA. By the way, the type of DNA in your mitochondria too is all maternal. The only genetic information in your mitochondria from your mom. The Christie, there isn't a thing that's the Christie. The Christie is the winding back and forth of these membranes. Membranes, it's all about that. And the matrix is just the very inside, Okay, so we'll talk more about mitochondria and what they do a little later on, but I do want you to remember a couple of things from here. Number one, I pointed out that they have DNA. The shape, they're kind of shaped like that rod-like bacteria I showed you, right? And number three, we know that mitochondria can divide inside your cell even if your cell's not dividing. So if you increased your aerobic workout, let's say, and you needed more mitochondria, you needed more energy in your muscle cells would reproduce as well, but inside your muscle cells, your mitochondria would reproduce, okay, to fulfill that need of more energy. Now, the way the mitochondria reproduce is interesting. It's almost exactly the same way as bacteria reproduce. They split in half. They split through just fission, we call it, just splitting in half. Okay, so remember that for the next through the next slide until I get to the slide after it, right? The DNA, they have their own DNA. The cell's got its DNA in the nucleus, right? This is another piece of DNA. They have a shape of a rod-shaped bacteria, and they divide when they need to divide. They divide the way bacteria do. Now, the other organelle that I'm going to mention, but come back to, is the chloroplast, where photosynthesis happens. Okay, so this is where basically carbon dioxide and water, typing this out. Oh, one more. Hold on a second. CO2 plus water yield 6, 12. Glucose. That's what I'm writing, right? So this is the opposite end of what's going on in the mitochondria, right? This is where we take an inorganic molecule, carbon dioxide, and hydrogen, or water, excuse me, and we make an organic molecule. We make out of that. Now, the energy is needed to do that, and of course, that's where plants need the sun for. Notice once again that we've got an inner membrane space, an outer membrane, an inner membrane. It's all about the membranes, folks. Membrane, membrane, membrane. It's got its own ribosome. It's got its own DNA. Oh, again, that's curious. It's kind of shaped like a rod-shaped bacteria. That's curious, too. And if a plant cell needs to make more chloroplast, it doesn't have to divide just the cells. It divides the chloroplast, very similar to the way the mitochondria do it, very similar to the way the prokaryotes do it. And you see what I mean in just a second, what I'm trying to get to. Now let's finish the anatomy here. These little green, they always look like Mentos to me. These individual sacs are called thylakoids. I think that's a cool word. And a stack of thylakoids is known as a granum. And a bunch of granum are known as grana. Okay. Now, notice when you look at the thylakoids, it's kind of like that winding thing, that crystal we saw in the mitochondria, and it's all membranes again. So the artist has drawn it, you know, as if it were cut open. You can see the chloroplast right here, and this is that's up in the upper right is an organism called Spirogyra. Cool name, huh? Got a neat name, Spirogyra. That's how the chloroplast wind around. This is an algal cell. Okay, what have I been getting at with the DNA, the shape being like a rod-shaped bacteria, and the fact that they divide that way? Well, you know, if you notice that, that wasn't lost on scientists many years ago. They noticed that, and they wondered where these very specialized organelles of the chloroplast and the photosynthesis came from, and they came up with a very compelling theory through a number of research and hypotheses. Back in the past, oh gosh, probably talking somewhere close to about 3 billion years or so, we think that an ancestor of a eukaryotic cell acting as a host engulfed a prokaryotic cell, a bacterial cell, specifically a proteobacteria, they call it. You can see it in the picture there. They engulfed an oxygen-using, non-photosynthetic prokaryote. Now, oftentimes you would think if it engulfs it, it would intend on eating it. But in this case, it did not consume the mitochondria. And the mitochondria, because the cell now had one, made the cell more efficient. Now, I don't want to say any more about that, how it made it more efficient, because we're going to talk about that in respiration when we get to that chapter. But by incorporating that mitochondria, man, I tell you, it changed the evolution of life on Earth. It was a significant event. The other one, or the other thing that may have happened, we believe, is that a chloroplast came about because one of these engulfing eukaryotic cells engulfed a photosynthetic prokaryote. Remember, I showed you a picture. I said there are some bacteria that can photosynthesize. So it's perhaps possible, this is called, by the way, the endosymbiotic theory of the origin of these two things, the mitochondria and the chloroplast. And the reason why scientists suspect this, right, or initially suspected this, for those three things I told you. They both have their own DNA, which is unusual because other organelles do not. They both have a similar appearance as a rod-shaped bacteria, or bacterium if we're singular, and the way they divide. They divide the way bacteria divide, just like splitting in half. Very compelling argument. Okay. And the questions on the endosymbiotic theory. Now, I didn't mention the benefit that the cell now has with the mitochondria, but my guess is, you could figure out the benefit from now having a chloroplast, right? What can you do if you have a chloroplast that you didn't before? Not hold water. But if you're chloroplast, what can you do? What's the function of chloroplasts? We just talked about that a second ago. What do chloroplasts do? Start calling on people. Let's see here. Amber. Both of you at the top. Amber Hopkins, Amber Sexton. They photosynthesize. Thank you, Marie. They make food. So that's an advantage, right? All you need to do to make food now is a cell. You don't have to go out and find it. You don't have to go get it. You can simply just wait for the sun to come up, which it's been doing for 4.5 billion years. So that's a pretty much of a guarantee. Yeah, exactly. Very clever thing for a plant or for a cell to engulf. Now, this will probably be our last one. Let me see, where are we? Yeah, this is known as a peroxisome. Peroxisomes are interesting in the fact that they really demonstrate the importance of cell membranes. Now, they're not really green, of course, but you can see the cell membrane around it. Inside of a peroxisome, there is an enzyme. We're going to talk about enzymes later on, but what that enzyme does is when you digest fats, what's called beta oxidation of fatty acids, when you digest fats, one of the things that's given off is H2O2. Not water, but hydrogen peroxide. And that is actually dangerous to cells. It forms what's called a lot of free radicals, and that tears molecules and cells apart. So what this enzyme does is it converts that hydrogen peroxide into water and oxygen. So it makes it non-toxic. That happens inside the peroxisome. The peroxisome, because of this concentration of hydrogen peroxide, is dangerous to the rest of the cell. It's stored inside of this peroxisome until it's converted into something less dangerous. But if there weren't cell membranes, that hydrogen peroxide material would leak out to the rest of the cell parts, like for example, these chloroplasts that are right next door, right, or the mitochondria, and it would destroy them. You see, so one of the things that cell membranes do that are really advantageous is, one of the functions is they compartmentalize things that are going on inside of the cell. So you could literally put something that is dangerous right next to these very sensitive organelles of the chloroplast and the mitochondria, and as long as you had this membrane that would keep them confined there, right? Isn't that neat? The analogy I always use is you could build a daycare for young children, obviously, next to a lion, safari exhibition, as long as you had a really, really, really good barrier, right? I mean, nobody in their right mind would do that, but you could see hopefully with my analogy. Okay? Let me just do this last one. Okay. This is what we're going to talk about when we get together next time. This is components of the cytoskeleton. The cell actually has a skeleton of sorts. Cyto means cell. I just think that's a neat picture. This is that fluorescent microscopy again. The blue thing in the middle is the nucleus, by the way. So we'll talk more about those when we get back together next week. Lab. What lab? Let's see. Next week is the 3rd. Oh, I see. So next week, we won't be doing a lab. Right? Next week's the 3rd. Yes. So there's no lab. We'll get caught up on lecture next week because we still need to finish 6, move into 7, and then start 8. 7 is pretty quick, actually, if I remember correctly. Okay, so the next lab we'll do will be the week of the 10th, and that'll be a lab that I have to send you. That'll be lab number 5. All the labs with asterisks that are in the syllabus are ones that I will send you. Notice there's 2 number 9 labs, by the way, 9A and a 9B. 9A is in your book. 9B is what I'll send you. So you really only need to print. Oh, 11 shouldn't have an asterisk. Shoot. You really only need to print 9A and 11. 9A is 9 in your lab manual. There's no 9A. Okay? Let's stop the recording.